Mitosis and meiosis are essential processes of life. Mitosis allows for growth, maturation, and replacement of damaged cells. Meiosis is important because it is the process by which gametes are made haploid in order for sexual reproduction to occur. Both processes begin with one parent cell consisting of organelles, DNA, and genes. Mitosis consists of three parts: interphase, mitosis, and cytokinesis. Cell begins with interphase, which is the longest part of the cell cycle, spending approximately 90% of its time in this part. Interphase is made up of three checkpoints, G1, S, and G2. However, when the cell is at a standstill, neither dividing nor preparing to divide, it goes through G0 phase. At this stage, a cell contains chromosomes. Following interphase, the cell moves into mitosis. Prophase is the first step of mitosis, where chromatin starts to condense into discrete chromosomes. Nucleolite disappears and nuclear membrane breaks down. At the end of prophase, the centrioles have migrated to opposite poles, chromosomes are moving to the center of the cell, and meiotic spindle begins to appear. Next step is metaphase, where the spindle forms and chromosomes align on the metaphase plate, which is an imaginary plane in the middle of the cell halfway between two poles. Each double-stranded chromosome is attached to a spindle microtubule at its centromere. Anaphase begins when the centromeres of each chromosome separate. Each sister chromatid now has its own centromere and is called a chromosome. There are twice as many chromosomes as there were during metaphase. Spindle microtubules attached to the centromeres cause chromosomes to move. Final step in mitosis is telophase. Chromosomes gather at the poles in telophase and begin to form daughter nuclei. The two daughter nuclei will be identical to the nucleus of the parent cell. The nuclear membrane begins to reform, nucleolite begins to reappear, and chromosomes begin to unwind and disappear from view. Final part of mitosis is cytokinesis, the division of the cytoplasm. In animal cells, the membrane constricts in a ring around the middle of the cell and eventually pinches the cell in two, known as cleavage furrow. As opposed to mitosis, which is a process of asexual reproduction, resulting in genetically identical and equal number of chromosomes in haploid cells, meiosis is a process used through sexual reproduction in which the number of chromosomes are reduced by half through separation of homologous chromosomes in a diploid cell. Meiosis consists of two parts, meiosis 1 and 2. Meiosis 1 is the separation of homologous chromosomes. In prophase 1, DNA coils and individual chromosomes become visible as tetrads. The nuclear envelope and nucleolite disappear. The spindles begin to form. In metaphase 1, spindle fibers attach to one side of each centromere and two homologous chromosomes attached to microtubules from opposite poles. Each pair of homologs then line up at the metaphase plate. Next comes anaphase 1, where microtubules of the spindle fibers shorten and pull chromosomes toward the poles, taking sister chromatids with them. Each pole ends up with a complete set of haploid chromosomes consisting of one member of homologous pair. During telophase 1, the nuclear membrane reforms around the daughter nuclei. Each daughter nucleus contains two sister chromatids for each chromosome, added to a common centromere. Because of crossing over, the two chromatids are not identical. Final step of meiosis 1 is cytokinesis, where the cytoplasm splits. The result is two daughter cells. Following telophase 1 and cytokinesis in meiosis 1 is meiosis 2, the separation of sister chromatids. Meiosis 2 has the same stages as meiosis 1, with the exception that during meiosis 2 there is a separation of sister chromatids, whereas in meiosis 1 the separation is of homologous chromosomes. In prophase 2, the nuclear envelope breaks down and new spindles form. In metaphase 2, spindle fibers bind to both sides of the centromeres. During anaphase 2, the spindle fibers contract and sister chromatids move toward opposite poles. In telophase 2, the nuclear envelope reforms around the new sets of daughter chromosomes. Finally, cytokinesis occurs again, resulting in 4 haploid daughter cells containing single chromosomes.